Hey everyone, it's Mr. Sinti, and today I'm pleased to be talking to you about the technique of Agarose Gel Electrophoresis. It's a great technique that we use in the lab for a variety of purposes. First off, the name agarose is it's a sugar, it's a polysaccharide, and we can turn it into a gel by mixing it into some electrophoresis buffer, and we can heat it and take it to a boil so that the polysaccharide dissolves. So once we make a gel, as you can see here, we can put it inside of a chamber and we can add some samples, often DNA, and then we can run an electrical current when we attach a power supply to it, and the electricity will separate the DNA or phoresis. So we can separate using electricity DNA samples through an agarose gel. And so it's a very important technique. It's one of the most important molecular genetics techniques that we'll be using this year. You can use it to analyze crime scene, DNA fingerprinting. You could separate individuals from one another, and that's what we're going to be doing in the lab. But we also use it a lot for other purposes. You could use it to identify, for example, a genetic marker. You can test to see if someone's a carrier for a particular disease. There's a myriad of purposes for agarose gel electrophoresis, so I hope you enjoy this discussion. Let's get right into it. And so let's come up here and let's try that. So agarose gel electrophoresis, the story for electrophoresis is going to begin somewhat obscure and sort of seemingly random, but trust me, it's going to have a good conclusion. And so I want to take you back to something that you might be familiar with. This is a simple bacteria right here under the scanning electron microscope, and these little orange guys are bacteriophages. They're viruses, and what they're doing is they're trying to infect the host bacteria, and what they do is their protein attaches to the outside and they inject their nucleic acid inside the poor bacteria. And so as it turns out, look at this. This is a great picture. Let me see if I can animate it. Inside here, the viral DNA is going to go inside the bacteria, and when that happens, the viral DNA is going to cause havoc to the bacteria. It's basically going to commandeer the cell. It's going to use the cell to make more viruses, and it's going to be Troubleville for the bacteria. And so here's a diagram of this. Virus attaches. Here's the bacterial DNA, circular chromosome. So what happens is when the virus injects its DNA, there's certain genes that cause the bacterial DNA to be destroyed, and then the viruses replicate and the cell lyses. Pretty brutal. So you might be, at this point, maybe, feeling a little sorry for the bacteria. And so this is where this conversation is going. You may not be aware of this, but the bacteria are not completely defenseless. You know, they have been living on the earth for over 3.5 billion years, so they've come up with a few tricks. So let me see if I can show you this. So if here's the phage attaching to the outside of the cell and it injects its DNA, what the bacteria can do is it has a very important class of enzymes called endonucleases. And these endonucleases are enzymes that they possess that can cut DNA. So this is what they do. They come up to DNA like that, they approach it, and they simply make a cut. And so when the virus, thinking it's cool, attaches to the bacteria to destroy it, these little sort of defense mechanism enzymes come along and go snip, snip, snip. Too bad for the virus. And so what I'm getting at is you might be wondering, well, okay, these are great enzymes that cut DNA, but hey, wait a minute. What about the fact that the bacteria has DNA? Why don't these enzymes sort of turn tail and attack their own plasmids or chromosome? Couple tangents here. They don't do that because the bacteria marks its own DNA or tags it with methyl groups in a process called methylation. So in other words, it marks it so these enzymes would not cut. So they only cut foreign DNA. Well, as the story goes, around the 1970s, biologists, I believe at MIT, started to see that these cutting enzymes called endonucleases, or sometimes called restriction endonucleases, can be very valuable. They could be removed from bacteria, and they could be used to cut DNA, any DNA. And so these enzymes were isolated from bacteria, and we can now use them to do a variety of analysis of DNA. And ultimately, we're going to be using them in Agarose Gel Electrophoresis in a moment. So to summarize, humans have taken these restriction enzymes or endonucleases away from bacteria, and we could use them to cut DNA. And so that's my point that I want to get at. And so they're restriction enzymes or endonucleases, meaning they're bacterial enzymes, they can cut DNA, and there's different ones. So they're called restriction because they don't just cut free willy, they cut at only particular sequences along the DNA strand. And so those are called restriction sites. And they're not very big. They're around 10 or so nucleotides in length. And they cut whenever they see that particular restriction site. So if you're following this, let me just sort of go like this. Here's a piece of DNA. And let's say that this particular restriction site is located right here. And here comes the restriction enzyme. So this restriction enzyme, endonuclease, is going to come along. It feels along the DNA and it recognizes this particular area and it's going to make a cut. So this is the result of it. It cuts the DNA. Now let's just set up another scenario. So this is DNA here. Let's say that you have another piece. It helps if I have the pen tool. Let's say you have another piece of DNA. This is DNA. This is DNA. This is, say, from person A and person B. And DNA is very similar between individuals, it's true, but there are differences between individuals. Of course, you know that. And so what's fascinating is one of the differences could be that there is a restriction site located here and one located here between person A and B. So if the same enzyme came, it would cut this person there, and it would cut, here's another enzyme, it would cut this person here, and this person here. Now, I know what you're thinking. You're like, well, if we could see this, this is probably in a little test tube, and the liquid's clear, seemingly clear liquid. If you were able to cut this person, and then this enzyme would cut this person, you would see this person would be cut into 3 pieces. This person would be cut into 2 pieces. So you would say, aha, there's the difference between person B and person A. The enzyme cuts differently, therefore there are 2 different individuals. But the trouble is, how would you ever see this? And again, it may not be clear to you right now what I'm talking about. I can appreciate that. But all of this is taking place in a little tube. So in other words, here's tube A and here's tube B, microcentrifuge tube. So you put the DNA inside here, you load it with a pipette, and then you add the restriction enzyme and it makes a cut. But it's a clear liquid. And then over here you put the DNA into this test tube and you add the restriction enzyme and it makes 2 cuts, and there's 3 pieces. But it's a clear liquid. You would never know. So it sounds a little weird, but I just have to point that out, that you'd never know that. So getting a little bit more particular about these restriction sites, they're often symmetrical, meaning that they're DNA is double-stranded, and so I'm just looking here at CTTAGG. It's often palindromic in a reverse direction. I don't know if you notice that, CTT. So it's palindromic, and then the enzyme is capable of cutting this DNA. at that particular site. It doesn't just cut anywhere, it just cuts in that particular site. So there's different restriction enzymes, and they cut at different restriction sites. And so one of the ways in which we're going to analyze the DNA and how it's cut by the restriction enzymes is by using Agarose Gel Electrophoresis. Now the thing is, the gel is made up of agarose sugar, and so if you can picture the gel, think of the gel as being sort of like a sieve, sort of like a colander where you're straining out your pasta. And so it's somewhat porous. Or you can think of a different analogy, maybe you're thinking of Swiss cheese. So it's sort of this gelatin material with there's holes in it. And as it turns out, it can separate DNA based on size. And so if you run an electrical current, if a current is applied to the gel, the DNA, since it's negatively charged, it'll migrate through the gel, and it'll sort of snake through these pores. And as it turns out, the smaller pieces will migrate the furthest, and if the DNA is really big, it won't go very far. And so the molecules could be sorted based on size. And so DNA fingerprinting, it's called fingerprinting because everyone has unique DNA. You could identify an individual based on the restriction cuts and running it through an electrical gel. So everybody's got different DNA, that is, except for identical twins. And so as it turns out, let me just sort of go over what we were talking about just a second ago. So if you had, sorry about that. So if you had this person right here, and then here's another person. So the DNAs of two different people, A and B, are about the same length. If you ran it through a gel, both of these wouldn't go very far. But if you cut this one here, and then if you cut this one here, and cut this person here, do you notice how this has 3 pieces and this one has 2 pieces? And so the way this is going to work is when this goes through the gel, let me sort of animate it if you don't mind. When it goes through the gel, the smaller piece will migrate very far, and this piece will go only a little bit of the way. Whereas in this over here, these 3 pieces are approximately the same size and they're kind of medium. They would look like this. So if you compared person A and person B, you would see that there's a difference between them. I'll continue with this. So this is going to be you. You're going to be in the lab, and you're going to take your micropipetter and your electrophoresis chamber and your gel power supply, and then you're going to add samples to the wells that you're going to be creating. Now the wells, remember, are created by this comb. And when you make the gel, it makes indentations. And so you're going to load samples of DNA. Perhaps crime scene DNA will be in lane 1, suspect 1, suspect 2, suspect 3, etc. Now, what's interesting is sometimes you need to use more than one restriction enzyme in order to identify individuals. And so sometimes that's what we're going to need to do. And so I like this diagram. This is showing you here's the DNA that's been cut with the restriction enzyme, so you're going to be loading it to the gel. And this is in the gel bed inside the chamber. Then we're going to hook up the power supply, and since the DNA is negative, it's going to migrate. So if I can animate this, it's going to migrate through the gel. So say inside this gel, in this tube, there's a big piece and it's cut like this. So there's a little tiny piece and a big piece. So the tiny piece is going to go really far, and it's going to go right there. If I'm able to do this, it's going to go right there. So that's the tiny piece. This one is here, and then this big fella is like right there. If you didn't cut it at all, say here's DNA that wasn't cut at all, maybe it wouldn't migrate very far at all. Because remember, the bigger the piece, the least traveled it's going. And so say this person right here was like that, and you cut it like this, and so the same as this, and so this person might look like this, just like the first one. So let's say that this is crime scene DNA. In other words, DNA that a criminal investigator found at the scene of a crime, and then this is a suspect. And it's like, ho, 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 it seems to match up. So perhaps this could be used to identify a criminal. Now, here it shows many cuts, and so this is kind of an elaborate one down below. So what's interesting about this is that when you add the DNA to the wells and they're cut with the restriction enzyme, the reason that it's looking purple is that we add loading dye to the sample so we can see what we're doing. Then when we turn on the power, the only thing you could see is the loading dye migrating through the gel. The DNA is actually invisible. We're going to need to then, when the gel runs for about an hour, then we need to pull the gel out and put it in a staining tray and add a chemical that attaches to the DNA. And then when we apply an ultraviolet light, it'll fluoresce. And then we'll be able to see the DNA. It's really cool. Looks like this. And so the DNA fragments, or restriction fragment length polymorphisms, will appear as these pink bands along the gel. It's really cool. And so we'll be able to see this, and it's pretty interesting. And so it's if the banding pattern matches up, then it's the same person. If it's different, then you're a different person. So these restriction fragment lengths will look like this. This is our ultraviolet light illuminator, and so you'll be able to compare DNA from crime scene with various suspects, and so it's pretty neat. Let me show you a little video of what I'm getting at here. It might be helpful. And so what you can see here is a piece of DNA, and this is a restriction site. And so the bacterial enzyme, a restriction endonuclease, will come along. Say this is a human DNA, or it could be anything. It could be any DNA. If it identifies the restriction site, this happens to be the name of the restriction enzyme, EcoR1. Sometimes it's just called R1. It'll recognize this particular sequence and it'll make a cut right there. Notice how this is complementary, and it'll make a cut right into this molecule. And so the enzyme's going to come floating in. So this is like in a test tube, and we're going to add the restriction enzyme, and we won't be able to see this, but it'll simply make a cut on one side and the other side. Let me just pause it. Now, this can happen a few different places, and we would never know it. And so it just looks like a clear liquid. We add DNA, we add the enzyme, but it's like, well, I don't know. You have to run the agarose gel in order to see the fragments. And so when you cut DNA, it'll create fragments. I hope that's clear. Let me show you this. Like, here is an example of DNA that has 3 different restriction sites. And so it'll recognize the sequence. And so these are the restriction sites. And so this particular person has 3 sites. And then, let me pause it here for a second. Notice here how this person has 3 sites and this person has 2 sites. And you're like, huh, I wonder how that happened. Well, if you look closely here, I'll let that play just a little bit more. If you look closely in this red box area, they're pretty similar. It's like this is an actual restriction site and this one's not. You're like, well, what's the difference? It's pretty much the same except for this base pair right in the front. And that's enough to where the restriction enzyme wouldn't cut it. It has to be just like this. Now, how can something like this have come up? Maybe a mutation. A point mutation could have changed this. And so this is how you would be able to tell the difference. Like, let this play. So what's really cool about this is that, here's one person, here's another person. So you put the DNA into one test tube and into the other. And then you add the restriction enzymes. And what's going to happen is these restriction enzymes are going to make a cut there, there, and there. And on this person, it's going to make only 2 cuts. So 3 cuts, 2 cuts. Pause. So if you make 3 cuts, you're going to get like this short fragment and a medium-sized fragment. And then over here, with only 2 cuts, you're going to get this big, long fragment right there. And so you wouldn't know it, again, in the test tube unless you ran an agarose gel. So here's a little simulation of the agarose gel. You pipette it into the wells, turn on the power supply, you let it run, so the current is pulling the DNA to the positive side, and then, whoa, look at that. you're going to get, in this case, 2 pieces, and over here you'll get 1 piece. And so you can then stain it, take a picture of it even using a digital camera, Polaroid. You could even scan it into a computer. But you would clearly see these 2 bands, as it's called, are different than this band. This big piece is different from these 2 pieces. Technically, these are called restriction fragment length polymorphisms, kind of a mouthful. But you can not only tell the difference between individuals, but this can also be used to determine a genetic disorder. And that's something that I want to sort of talk about now. That's kind of a neat thing. Let me come over here. Say, for example, somewhere in your family there's someone who has sickle cell anemia, and you were wondering, I wonder if I am a carrier. I know that you may know that you're not, you don't possess sickle cell anemia, but you might be a carrier for it. So how does one know that? Well, as it turns out, we know the gene for this beta globin area. I don't want to get too detailed, though I could, but there's this one area on the gene for hemoglobin. And hemoglobin is if you recall this from a previous discussion, if there's a mutation in the gene for hemoglobin, that's what this big purple area is, it could result in sickle cell anemia. And so what's interesting about this is, we'll talk about this a little bit more, but you have a piece of DNA from your mother and from your father. And so as it turns out, here's the normal or wild type right here. And so this is the correct sequence. And so this red is a restriction enzyme. And so it'll make a cut there, 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 and there. And it'll produce a large fragment and 2 kind of smaller pieces, if you can see that. Over here you can notice that. Here's the large fragment. Let me make it very clear. The large fragment is right there. And these 2 pieces here and here will be these 2 pieces. See, the 201 is right there and the 175. So this is normal. If you were normal, in other words, your mom and your father gave you 2 normal pieces of DNA, this is what you would look like. If you were sickle cell, in other words, what happens is, because there's a mutation in your DNA, one of the restriction sites, this fella right there, this one is no longer going to be cut. And so as a result of that, there's only a cut there, there, and there. So there's 3 cuts, since we have linear DNA, resulting in a big piece. That one's the same. So notice here. But notice over here, instead of 2 little guys, there's 1 medium guy. And so there's a medium guy there and a big guy. The medium is here and the big is here. Notice the big one didn't migrate. So gel electrophoresis can be used to determine whether or not someone's a carrier for a particular disease. You may not be showing it. We'll come back to that topic. It's really important. But I thought I'd mention it because, in other words, all you need to do is go into a lab, have your DNA extracted, maybe from a cheek cell, and your DNA can be analyzed using restriction endonucleases, and then gels can be run, and you can determine whether or not you're a carrier. I think that's pretty awesome. I think it's pretty awesome that you're even following this conversation. And so you should be impressed. And so finally, let me just show you here, in conclusion, what we're getting at is, in the lab that we're going to be doing, you're going to be pipetting. So we're going to be using the micropipetter. And just a little review. To pipette, you go down to the soft stop, I should say, and then you're going to pipette your DNA. Now this is DNA that's been cut with a restriction enzyme. Sometimes we can add them, sometimes they're previously cut or digested. So you're going to then take this and then come over to your gel. You're going to go through the buffer and you're going to pipette it. You're going to push it all the way down until it fills the well like that. And then you're going to change the tip. It's very important. If you don't change the tip, you might be contaminating the samples and send an innocent person to jail. You don't want to do that. So there you are, loading the gels, and then you're going to put the top back on, and then you're going to turn on the power supply. You're going to run the gels, and we're going to find out who was the suspect at the crime scene. So I hope you enjoyed this brief video on the joys of Agarose Gel Electrophoresis. Thanks for watching.